Buenos días a todos y todas. Bueno, iniciamos hoy el último módulo de este curso de citometría de flujo. Vamos a contar con dos disertantes internacionales, expertas en el tema, así que esperamos nuevamente que puedan aprovechar estas disertaciones como lo hicieron en el día de ayer y antes de ayer. Les pedimos que al final de las dos disertaciones no se vayan, que Guillermo va a compartir con ustedes también unas direcciones de repositorios y de sitios a donde pueden conseguir algoritmos, de modo que no se desconecten en el último momento, así pueden acceder a eso también. Ahora los dejo con Guillermo, que va a presentar a nuestra primera disertante, a quien agradecemos desde la Sociedad Argentina de Inmunología, muy en especial por haber aceptado participar de este curso. Muchas gracias.
Hello everyone. I'm very proud to present Sophie Van Gasten, who has generously accepted to give us a presentation over computational cytometry. Sophie Van Gasten received her PhD in computer science engineering from Ghent University in 2017. During her PhD, she developed machine learning techniques for flow cytometry data, including the FlowSum algorithm. She is now working on tools for computational cytometry in clinical studies during her postdoc in the SAIS lab, VIV Eugen Center for Inflammation Research. Since 2018, she is an Isaac Ingram Marlowe Scholar. So it's a great pleasure to have you here, Sophie, and we are willing to see your presentation.
Thank you very much for the introduction. It's a pleasure to be here. I will try to start sharing my screen now. There we go. So if all is well, now you can see my slides. And I will talk to you today about computational flow cytometry, like was introduced, which is my main topic of experience. I will talk first a bit about the different tools which are currently available in the field and which might be interesting for you to check out, to have a look at. Then I will shortly touch upon which platforms you can actually use to use these tools. And then I will end with some extra considerations you should take into account when you're considering using these computational tools for your research. So I'll start with the different tools which are available. So currently, I don't think there's many tools available for data generation. This is still the work you do in the lab. But once you've measured your data, there's actually lots of algorithms that can help you with all the following steps of data cleaning, data visualization, and then the final more statistical analysis. And there's like lots and lots of algorithms out there. I'm sure you could come up with many more names. And I will give a short overview of these. So I think visualizations are always crucial when working with data. This is what creates insight, what makes sure that you know what's going on with your data. And even in the most traditional way of working with flow cytometry, you will be using these scatter plots to look at data. Even when you go to more computational techniques, these scatter plots remain very valuable. You can also take some different scatter plots, for example, looking at time versus a certain marker and noticing if there's any strange things occurring during the measurements. You can also then move to dimensionality reduction methods, because here you can only look at 2 markers at a time, and now the number of markers that are measured just keeps increasing and increasing. So only seeing 2 of them at a time gets a bit limited, and it might be interesting to combine information from all of them in one 2-dimensional plot. You could also look at clustering results. Here I show an example of our FlowSum technique, which is of course what I'm most experienced with and which I will go into more depth on later. And then you also have other alternatives, for example, trajectory inference. And there you will also really use all kinds of visualizations to make sure you understand what's going on in your data. Very shortly, but a little bit more detail about dimensionality reduction. I think the best known dimensionality reduction methods in general, not for flow specific, but just in the computer science field at all, would be PCA, principal component analysis, where you're actually looking for the direction that explains most variation in your data set. And you could also apply this on flow data. I have an example figure here. But as you might notice, like in this cloud over here, there's actually a lot of colors mixed up. So while there are some big variances between some cells, some others are just differing in only a few markers. And then a PCA separation won't split them up very well. So therefore, PCA is actually not that suited for cytometry data. What's typically used is t-SNE or v-SNE. And here you can see that all the islands are separating much better. And that's because it takes a more local approach. So it will only try to keep the nearest neighbors close to a cell. One thing I want to remark here, if you're using these techniques, I would really recommend to also have a look at this paper by Annabel Kina, where she's really going into detail about optimizing the parameter settings for this t-SNE plots, which can really make sure you get the most out of this technique. There's also some more recent ones like UMAP, which tries to find a kind of in between both capturing really the local things going on and then the more global patterns as well, which typically results in these islands which are a bit wider spread out. It can also be slightly faster than running a t-SNE. And then there are also some more recent and exploratory methods, like for example, this embed sum, which actually builds on the flow sum ID. It also uses a self-organizing map to map the individual cells in a two-dimensional space. So lots of options out there and certainly very helpful if you want to get an overview of your data. However, one remark that's very important to me is whichever of these techniques you use, they don't give you a clustering, they don't give you cell populations. So even though you can see things here, it's unclear whether this should be a separate island or maybe this is a full island. So it's just giving you new 2D coordinates. And if you want to start doing statistics, you will need to group the cells in some way to be able to compare them between samples. One way you might consider this is just to automate your gating. So you have done a manual gating before, and you say, okay, now I have to do this for my 3,000 other samples. You could do it by hand, but it will be very annoying. So you might consider maybe I can use a computational tool for this instead. Next to these clinical studies with lots of samples where you really know what you're looking for, there's also sometimes some pre-gating steps, I would call them. So where you know, okay, I want to do this unbiased exploratory thing of my data, but I do know upfront that I'm not interested in debris. I'm not interested in dead cells, for example. I don't want to do an exploratory thing of all my dead cells. So there as well, it might be interesting to first just reproduce some known gates as a starting point. So if you want to do this with computational tools instead of doing it in the traditional way manually, I would suggest you could have a look at this OpenCyto framework. It's proposed by Greg Finnack, where you can really kind of build a whole hierarchy of which steps you would take, actually very similar how you would do it in a manual gating. But then you can define how each of these gates are made, and there are all kinds of tools for you to actually implement that. One tool I've used myself quite often is flow density, and this one will actually look for the optimal split for a specific marker. So it looks at one dimension at a time, or I think it can also do two at a time. I typically use it for one at a time, and it will just look for the optimal split. And if you have something like this, I'm really sure that the algorithm can do it just as well as anyone would do it by hand, and it will just work perfectly for all your files. If you have a situation like this, where the the split between the two populations, between your negative and your positive cells, is a lot less clear, it might get a bit more tricky, both for us to do it manually, as for the algorithms as well. So in that case, we had an example here where we were just looking for like the main populations that you would see in a very broad panel. And then you can see, if we compare here the manual percentage determined on the x-axis versus an automatic percentage determined on the y-axis, like there's a perfect correlation. This determines it almost one-on-one. I would fully trust it. However, if you have some more difficult markers, some smaller populations, a more complex panel, actually for some populations, the method will fail. And so that's something to be careful for. This is really something to use for these first gating steps or for very well-defined markers. In other cases, you need to really double check the results it's giving you. One option, if it's not giving you the optimal results, is that they did this follow-up method called Flow Learn, where it will actually try based on, I'm really sorry about this, okay, based on kind of the distribution they see, they will try to translate it to other samples. And this way you can still say, okay, for one sample, I have the knowledge that the split should be in a certain position. And even if it's quite hard to determine, I know it's due to some background knowledge I have. And then the algorithm will try to translate this to all your other samples and move it appropriately. So that can be very helpful then as well. This is a thing I already mentioned. So the next kind of tools I want to talk about are clustering tools. And these clustering tools, they're different from dimensionality reduction, because in dimensionality reduction, you typically can't really quantify what's going on. You just get a nice visualization, which is helpful, but not something you can further compute on. It's also different from this automated gating, because with the automated gating, you would still define like, these are the gates of interest. I will first look at these two markers against, then at those two markers, and so on. Where in clustering, you're more looking into this explorative setting where you're not so sure what to expect. So where you just say, okay, I want to compare everything with everything, and I want to know what's going on in my data. One small remark, it's also something maybe a bit personal for me, but I would be very careful about calling this kind of analysis completely unbiased. Of course, there are still bias in there. You choose which markers you measure, also in the numeric or in the parameter choices of the algorithms. It's very easy to have some bias there, but it's for sure a more explorative setting than just looking at what you expect to be there. Of course, the clustering algorithm I have the most experience with is FlowSum, because that's the one I helped to develop, and I've also been using a lot in the last years in all kind of analysis pipelines we have set up. The main idea of FlowSum is that it's actually a two-level clustering. So first, we over-cluster the data. We make way more clusters than we expect there to be different cell populations. But the advantage of this is that we can do it with this very fast self-organizing map. So we just split up way too much, but then we can use this very easy technique. And this results in all these different circles that you would see here, which actually each of them represents a group of similar cells. And then what kind of cells are in such a circle, you can see by the little, it's star diagrams, star charts we have. So it's not a pie chart. The size of each part and the color and the angle always stay the same. The only thing that's changing is the height of each of these pieces of the pie. And then, for example, if we look over here, we can see here the red and the orange part are big and all the other ones are very small, which means these cells have CD19 and MHC class 2, so these will be B cells. While if I look over here, they don't have the orange and the red, they have the green part. This is the CD3, so this here will be T cells. And this also shows you the overclustering, because actually very many of these will have a very similar similar pattern, even though they are separate circles, so separate clusters. In a second layer, then, we apply a more sensitive hierarchical clustering. The disadvantage of this would be, if you would do it on the original data, it would be very slow. If you do it on 100,000 or 1,000,000 cells, it would take ages. However, we are only applying it on the previous clusters that we've generated. And that's then the background color you see. And if this is only applied to like 100 clusters or 200 clusters, that's really feasible to compute and goes very fast. So then we can take this more computationally intensive algorithm. And there we will see, this is the background color, that actually all these nodes which had the very similar patterns are now clustered together. And then we can see that this corresponds really nicely with how these cells were labeled manually. So in the traditional gating, we mapped it onto this tree. And then you can see, for example, the T cells, this corresponds nicely one-on-one with this automatic defined cluster here. So first, very fast, but not so precise, so we make very small clusters. And then in a second step, we do the aggregation again of grouping them back together until they correspond nicely with the populations that are actually of biological interest. Our algorithm was then also taken along in this benchmark study done by Weber and Robinson. And there we got actually a very nice result, because on the one time here, they evaluated the runtime. So of course you want it to be as fast as possible. And on the other hand, they also evaluated the mean F1 score, which basically tells you how well it corresponds with the manual gating results. So how well it captures the things that are biologically known of interest. And then we could see that FlowSum was not only one of the best performing algorithms, but also one of the fastest ones. So we would have expected more of a trade-off there, but actually FlowSum came out very well in this case. As you can see here, the FlowSum Pre, this is the one without the second step of clustering, there would get a less good F1 score. So it doesn't correspond so well with the manual gating, but it's what makes us fast in the end. An important remark, if you're looking for really rare populations, very small ones, there might actually be algorithms out there who can do a better job. So with FlowSum, the thing you would do is you would increase your number of clusters to try to get a more detailed level captured, but then there might actually be some algorithms better suited. However, it will be at a trade-off of taking quite a bit longer to run and to compute. Maybe one last remark about this runtime, why I think developing a fast algorithm is so important, is because this also gives you the opportunity to maybe run the algorithms a few times with a few different parameters and really explore what's going on, which is just something you wouldn't do if it already takes 5 days to run the algorithm. You won't say, okay, now I will do it 3 more times. No, you will just take your results and go with it. Okay. Once the data is clustered, you will get something like this. So what we have here is a heat map, and then we have all the different samples that were measured on the y-axis, and all the different cluster percentages on the x-axis. And then this kind of overview table, you can basically get it from any clustering algorithm you would consider. You could even make it with this, the percentages of your manual gates, of course, as well. So we typically use FlowSum to get to this point. If you prefer another method, that's also totally fine. And then you can take this for all kinds of extra steps you would want to apply. You can do dimensionality reduction, this time not of the cells, but of the samples, to see which of your samples are really similar to each other, if there are maybe groups of patients appearing, these kind of things, very similar then with the clustering ID as well. You could start doing statistics, comparing 2 groups of patients, maybe seeing which clusters or cell populations are statistically significantly different between them. You could maybe do some predictive modeling, try to make some diagnosis of a prognosis model based on previously labeled samples, where if you would get a new patient, you could then give a prediction for them. And then also biomarker discovery is, of course, a thing we're very often looking for, where once we have these predictive models, we will see which clusters, which cell populations were now actually most of interest into making this diagnosis, and maybe they could then be very suited as new biomarkers in making these models more accessible in the end if you actually want to use it in a clinical setting. I have one small example here of what a visualization like that would then look like. In this case, there are 3 groups. It were 3 groups of mice. We had the wild type mice, a heterozygous knockout and a homozygous knockout. And here the size of the circle is actually adapted to the number of cells that were present in that cell population. And then you can see immediately that here in this knockout, you have a huge increase of these cells here in the middle branch, while because we're looking at percentages, everything else seems really small. In the heterozygous case, this effect is like there's still some increase, but it's much less strong than it was in the full knockout. And actually, there's still one significant decrease here, and these were the cells of interest for the researchers of this model, so this was nice that this showed up. But one thing that for me was very interesting when getting to this result is, okay, they knew in the full knockout there was this huge neutrophilia, and this is something also if they would have looked at their percentages of their manual gating, this was the main thing that came out. The neutrophils increased a lot, and then everything else seemed to decrease in comparison. Well, actually, if we look at the tree here, there's still quite a part that's still increasing, and which was just not labeled in the manual gating. So you might kind of overlook this kind of things. When the researchers then looked further into it, these seem to be neutrophil precursors, so they were not explicitly gated in this panel. And actually, this kind of exploratory approach can give you these extra insights, which you might miss if you just look at the manual gating, only looking at the cell populations you expect. I will just go over some other tools that you could use in these situations as well. One of them is called DivSit, and very recently they actually also had this SenseIt extension of this. And the idea here is quite similar to what I explained, that you have your cells, you do your clustering, and then in the end you get also this big matrix with the clusters and then the abundances. Something we're also often looking at, which I haven't talked about before, is that of course you can also look at MFI values for these clusters. And then the main thing happening in this DivSit paper is that they actually will use more advanced statistics, while we often just go for a simple Wilcox test to compare between 2 groups, for example. They will have some more advanced statistics that they propose to get really the most out of your data. Citrus uses a bit of a similar pipeline, but they use a different clustering approach. So DivSit was also using FlowSum. Here they use this hierarchical clustering on the cell level immediately, which means that while this tree might look a bit similar, it's very different, because here in our previous trees that I was showing, every cell or every circle presented a different group of cells. Here it's actually the middle one will present all the cells, and then it's split into 2 parts, and then it's split again further and further and so on. So they compute it a bit differently. Once they have all these clusters determined, they will use a regularized regression model for the statistics, and in the end they can also come up with the main differences, for example, between 2 groups of patients. So again, similar to the things I was describing before. Cell CNN takes a slightly different approach, because in the approaches I've talked about so far, you would really first do the clustering and then apply some statistics on that. Cell CNN tries to really incorporate all of this into one method. So here you have your individual samples with all the individual cells, and then here actually the grouping is happening, so all the different patterns of markers are detected that could be there. And then you have your output layer with the final model prediction, so detecting the differences between different groups of samples. For example, different disease subtypes or whatnot. And then actually all of this is optimized together during the learning phase, so that he's actually really trying to find exactly those populations which are informative for your final label of the samples. And by not having this in two separate steps, but all of this in one model, this could also give some advantages, of course. A slightly more recent method is FAUST. That was recently proposed. I think one of the interesting things here is, so it will actually try to find all these splits of interest. It will also do it in a kind of exhaustive way, so not only looking at the splits you upfront decide this will be of interest, but kind of looking at all things that are going on. And it will both have some steps that are approached for every individual sample, and then some steps that bring it all together again across the samples. And this is something typically when we do a flow sum approach, we will just take all the samples together, make our model based on that, define the clusters based on that, and then afterwards split it out again into all the different samples, which makes that if your boundaries for your gates would really be different for every individual sample, if in a manual gating you would be shifting them around for all the samples, this approach will not be very suited. Well, if you have an approach like FAUST, which takes really the per sample computation first, this might be much better suited there. And then in the end, they also have the step where once they have the clusters, they will look what is now occurring across multiple of the samples. And in the end, you also end up with this kind of matrix like I was showing before. And then a bit similar to this idea is actually this also very recent paper called FOPO, where they also take this idea of maybe we should not first merge all samples together and then build one model based on that. Here they actually take all the individual samples and they would potentially make, for example, a flow sum model for each individual sample. So then you have lots and lots of clusters, and then you apply a meta-clustering step across the samples. So that's then the point where you link things between the samples, make them comparable. And then they include also some nice feature selection steps to really figure out which clusters are then of interest to, for example, classify between two groups of samples. Okay, so that there are lots of tools. I very shortly gave the main idea of these. You might be very interested in them. You say, okay, I want to use this on my data. How do you go about it? Which platform to use? And here, of course, it's a consideration typically between these more graphical user interface tools. So GUI stands for graphical user interface. Which is nice because you can just point and click at things. They are very easily understandable, or quite easily understandable what's going on. For example, Flojo, Cytobank, FCS Express, I'm sure there's more. I'm not affiliated to any of them. And actually, the most popular algorithms are typically made available in them. I think all these vendors are making a huge effort to really make sure their tools are up to date with all these recent developments in the computational cytometry field. And the advantage is, of course, also if you're using one of these tools already for your traditional analysis, it can be very easy to integrate also these more computational approaches. The alternative is that you would consider writing some scripts. The disadvantage of this is that it's quite a learning curve to get started with them. You don't have all the nice buttons everywhere. You need to start writing lines of codes to get started with it. But once you get over that hurdle, it has many advantages. It typically offers you quite some more options. You can make different visualizations than the ones that were previously in mind, and that can really be suited for your specific use case. The newest tools, an algorithm that's just published, it will probably have code available, but will not be immediately available in this graphical tools. So for me, of course, I'm mainly working in R. I think for the flow field, this is currently still where most of the tools are developed, so also where it's easiest to build on works of others. I know there are some tools available for MATLAB and Python as well. So for me, it feels like it might be worth to learn it. Of course, if you already have lots of things to learn regarding lab work, all the biology, all these things, maybe this just goes one step too far. And then it can also be very valuable to explore these algorithms in the more traditional analysis setting. Yeah, this is just an overview, like I said, and I think this is even already a bit an old overview, so it might be extended already. These tools have quite some algorithms available in them. Okay, and then I get to the last part of my talk, which are some extra considerations when you would consider using these tools. First, a small recap, actually. Choose the right tool for the right job. So I mentioned lots and lots of tools, and you always need to think a bit, okay, in which situation am I now? What should I use? So are you looking for a very specific cell type, which you know very well, which is well characterized? Or are you more in an explorative setting? Do you want to discover what's going on in your samples? This will decide whether you go for this more supervised automated gating approach, or if an unsupervised clustering approach is better suited for your situation. Do you want to get a visual overview of your data, or do you really want to quantify? In many cases, you probably want both. So then you should both apply a dimensionality reduction and a clustering tool, and realize that they're not the same, that they're, oh, I'm really sorry if these pop-ups are showing up. This is not supposed to happen. I hope you didn't see that. Okay. So there's a choice between dimensionality reduction or clustering, or like I said, they're typically quite, just use them both and realize what is doing what. And then if you want to quickly try something, the commercial software is probably the easiest to get started with it. If you want to make like some really specialized figures for a publication, really apply to your specific situation, it might be worth it to do the programming effort or collaborate with a bioinformatician who can do that for you. And then one of my main points I also wanted to make is that we have this saying in computer science that goes garbage in is garbage out. And that just means that quality control is crucial. If you put random data, random numbers into a clustering algorithm and you ask it for clusters, it will still come up with something. It doesn't necessarily mean that this would be in any way biologically relevant. So while I would already strongly suggest you do lots of quality control in the traditional flow setting, for a more automated approach, it's even more important. Because in an automated approach, you're not looking for hours and hours at these 2D scatter plots like you might be in a manual analysis pipeline. And then it's important that you also reproduce the steps that do this quality control. Just as a small reminder, for a computer, 1 means 1. It's not like, oh, but for this sample, 1 means something is slightly bit different than for that sample. No, if that's the case, you really need to clarify that for the computer. He won't make that up by himself. So one thing there is to check your measurement time on your individual samples. So have a look that no strange things happened during the measurement, and then also to check for batch effects between the different samples. I have some examples coming up of this. Batch effects are a very important thing. It can, yeah. If you're working with manual gating, you will probably just shift your gates around to adapt for that. And it's important not to forget about it if you're doing an automated approach. And right now, I'm afraid I don't have a full answer yet on how to solve it, but at least pay attention to it. So here I have an example of an individual sample. This is one sample that was measured. I have the time on the x-axis in all these plots. On the first figure, I have the actually the cell count, so the number of cells measured during a certain time frame on the y-axis. And then here on all these other ones, I have the expressions that were measured. And as you might notice, something happens here at this point, and then afterwards all the expression values are very strange. If I put the sample like this into a clustering algorithm, I'm sure it will find very interesting cells here. What happened in practice, I don't know, I don't have it indicated. What happened in practice is here the tube ran empty, and this is just air measurements with all kind of random noise appearing. And even though it might show up as something very relevant, this will probably not what you're biologically looking for. So this is just something to be aware of. Even if you're not using any computational tools, you can still make these plots to look at what's going on over time and try to notice this kind of things. Of course, there's also many tools available, or at least several tools available, which can help you with this. We used to use Flow AI quite a lot, which will basically do 3 checks. It will check if your flow rate is stable, it will check if your signal acquisition is stable, and then it will also check if there's any values out of the dynamic range of the machine. It will filter the cells in case there's any issues, and then it can export new files in which these problematic regions are removed. Actually, we're also currently working on a new tool called Peacock in our lab, which tries to improve on this a bit further, which will be soon sent out and then hopefully go through a smooth reviewing process. But the tool is already available on GitHub at the SciSlab GitHub. So if you're interested in trying it out, you can already go there and give it a try. The next thing, once you're sure that each of your individual samples is trustworthy, is to have a look at the stability between different samples. So what I'm showing on this figure is actually on the x-axis now, it are all different samples. And on the y-axis, it's still the expression that was measured. And if you're looking at this for a bit, you will notice there are 3 groups, right? I think this is one where it's pretty clear that here in the middle, we really have a very different pattern happening than on the other 2 ends. And actually, I think in most markers, things are going on. There are clearly 3 groups. There's, of course, 2 options. If it could be that it's just like 3 biologically different groups, like maybe we had with the example with the mice beforehand. This could make sense. Maybe in a certain group of samples, indeed, there is less signal of a certain marker, whatnot. However, if you notice this and the way the samples are ordered is just by measurement day, and this is day 1, day 2, day 3, then it doesn't make sense. Then it's just technical artifacts, and that's really an issue you should try to avoid. So I just want to stress how important it is to check for these batch effects, especially if you're using automated analysis tools. Make figures like this one. You can also do a kind of PCA overview of your samples, these kind of things. And test the things which you think would make sense. So for example, ordering them by the time when they were measured is very important. If you were measuring maybe on 2 machines, you want to make the plot like ordering them first all of one machine and then of the other machine. Going through different centers might cause a batch effect, all these kind of things. Also very important is try to think about it in your experiment design. If you are working with 2 groups of samples, don't measure all of one type one day and then all of the other type 3 days later, because then you have no way of knowing, okay, this is a technical difference or this is the actual biological difference of interest. So try to make a good experiment design that mixes your samples as much as possible over the different batches that you will have. And if possible, also take some control examples along. So once you have this, you can still try to kind of rescue it if there's only a limited shift happening. Maybe just some rescaling could be sufficient. However, then extra controls really might be necessary, having a sample aliquot that you measure on each of the days where you know there should not be any biological differences. So this is the way you can confirm this. And just realize if you're applying these normalization algorithms, there's really always a bit of a risk that you might also remove some of your biological signal. So we published the cytonorm algorithm kind of in parallel. There were also some different proposals. I think right now it's still an active field of research, and I think that's something I want to focus on in the future as well, really exploring these methods more in detail, seeing in which situations which method would be the most appropriate, the safest to use, and so on. So there are tools out there, but always be very careful and always, like I said, visualize as much as possible to really know what's going on with your data. Okay, and then I really get to the end. I think this will leave us some time for questions, so that's nice. I have a few resources that I just wanted to point out. If you're interested, or if you're a FlowJo user and you're interested in these tools, have a look at FlowJo Exchange, in which they show a lot of different plugins they have available. If you're willing to make the jump to actually start programming, this is a very nice resource about getting started with R programming. And more specifically, if you're then wanting to start doing cytometry things, have a look at Bioconductor, which basically just collects all kind of different libraries. They have a specific tag called Flow Cytometry, where you will see there's lots of packages available. And if you want to have kind of a recap of all the things I said and all the different types of tools that are out there, I will also make some small advertisement for our paper that we wrote in Nature Reviews Immunology, which also gives a kind of overview like I did today. And with that, I'm at the end of my presentation, and I'd be very happy to answer any questions.
Thank you very much, Sophie, for this excellent and comprehensive presentation of the computational cytometry and the status of this technology today. Waiting for the questions to arrive, I would like to ask you about one very leading edge technology that you mentioned in one part of your presentation, the application of CNN and a CNN-based algorithm. Maybe we are aware about CNN in images and these features that are like vertical lines or horizontal lines, but we can't figure out what are those patterns that you mentioned that these approaches see in flow cytometry data. What are those filters to be learned?
Yeah, Okay, so the filters that are learned in that approach are actually really corresponding to certain patterns of markers that you would have. So basically, it will kind of learn that there are often cells presented which, I don't know, are CD3 positive, CD4 positive, and then we have cells which are CD3 negative, CD4 negative, but they are CD19 positive, and so on. So actually the filters that are learned are these kind of marker patterns that are discriminated. And then they are in their aggregation step. So that's also something typically applied on these images, that you take a few pixels together and group them. In this grouping step, then they will actually kind of count how many of these different patterns are appearing. So that's different from how it's implemented for the images. And this way you get kind of the idea of what I was showing in this matrix beforehand, where you had all these counts for the different populations in your specific sample. So in the filtering step, they're really looking on like the single cell level, which marker patterns are appearing, and then they group them together, they aggregate them, and that's then what they use for their final classification. I hope this clarifies it a little bit.
Intuitively, it is outstanding because it's like placing human eyes inside the sample and looking in detail what's going on there with completely new approach.
Exactly. And so I think the main thing about it is the fact that it's in like one optimization model. So it's really also trying to optimize which cell populations it detects, to already know that you want to find the ones which are optimally different between your two sample groups of interest. Rather than having it in two separate steps, where first you're just looking for every possible population and then finding out what's statistically of interest.
Okay, I'm sure Ariel has some other questions. Thank you very much.
Questions. Thank you very much for your very good presentation, Sophie. Taking account of disadvantage of manual gating, especially in a very big number of markers, why do we have to validate the performance of clustering algorithms from the manual gating from expert people? Do you understand what I mean?
So I think, let me recap the question if I understood it right. If we have such large panels and we say, okay, we don't really trust the manual gating anymore, can we still use it to evaluate how well the clustering algorithm is working? Is this what you kind of meant?
Yes.
Yeah. Okay. So my response there would be is, indeed we might not like fully trust the manual gating and say, okay, it has limitations and whatnot. But you still expect a certain level of correspondence. If it's like not corresponding at all and coming up with totally random different things, probably it's also not that relevant. We have biological background knowledge. We don't need to throw it all overboard. So you still expect a certain level of agreement between them. I would agree that you should not use it in that case to maybe go to the level of saying, okay, this algorithm gets a score of 0.9 and this algorithm gets a score of 0.92. Yes, this is such a small difference there, you don't know. Like, I would not do it for the actual ordering of the algorithms, but just checking that things are making sense as a kind of sanity check, that there is a certain correspondence, that's still very valuable. Additionally, you would go to other methods of evaluation. So there are some clustering evaluation methods that just work on the clusters themselves without an external label to compare with, which can be helpful. And then you can also still say, okay, my algorithm is finding this cluster, which I did not expect. I will start inspecting some visualizations, looking at scatter plots, all these things to get more insights into what's happening. So then I think it's and the idea of combining these three different approaches to really get a full insight on what's going on.
Okay.
Another question is, do you recommend to use FMO controls when using FlowSum? If yes, how do you recommend to apply it?
It's a very nice question. I think in general, I would say, yes, I recommend using FMO controls. How to apply them when using FlowSum is slightly more tricky indeed, because our algorithm is not really like made for applying that internally. So I think one way we sometimes integrate them both is where we do the clustering just in the normal way, like I explained in the talk, and not using the FMOs, just on the actual samples of interest. But then we go, instead of looking at MFI values, we want to look at the percentage positive for each marker in each of the clusters. And there we can then use the FMO to find a good threshold for what we call positive. And in that specific situation, like typically you have some more linear lineage markers, which kind of define your main populations and which are also reasonably well defined. So for those markers you use for the clustering, they are capturing your main cell populations. And then you can use the FMOs for these more tricky functional markers and evaluate there the percentage positive in each of your clusters. So instead of using the cluster to really also separate all of these possible functional things, especially because there you typically have these more gradual patterns appearing, which are harder to split up, there then you can use the FMO to decide what you call positive in assessing each of these clusters. We are kind of considering if we can really take it along in the like in the distance metric that's used in the algorithm, but that gets a bit more complex and is still very experimental. So that I'm not sure I would like immediately tell everyone to do it that way. Yes.
Okay. Well, one question would be, you talk about trajectory inference, and we've seen FlowSum as a minimum spanning tree that sometimes very similar clusters could be placed far apart, and there are other algorithms that try to solve that way. How should we interpret the distances in the plot of a FlowSum diagram?
Yeah, a very good question as well. So in general, like you say, some parts of a FlowSum tree, sometimes you can almost think of them as a trajectory, where you have the gradual changes happening over a certain branch. Still, I would not say it's really like, if you know you want to go for trajectory inference, I think there might be some better suited methods, and I would not necessarily recommend FlowSum. I think overall, so the important thing to realize when using FlowSum is in the tree visualization, he uses this minimal spanning tree, which means that he will try to connect everything. That's already one big thing. So everything will be connected somewhere, even if there was not an actual trajectory developmental pattern between them. But also he will try to just connect them in a minimal way, so where the differences between the nodes which are connected are minimal. This means that for sure, if I take one node and I like extract it from the tree and I would put it somewhere else, it means the distance will be slightly bigger. It will not be minimal anymore, but that doesn't mean it's necessarily huge. If you would have a loop in the data somewhere, let's say your data is something like this, it will split it up somewhere because there can't be loops. It's a tree. That's just how the algorithm works. And then like these 2 points are completely at a different end of the tree, even if they were very similar, because you originally had a loop in your data. So you really don't have this guarantee about it. And I think that's you know that the things which are connected are very similar, but you don't know that things which are not connected are not similar. That's an often made mistake in the reasoning about these trees. And so be careful, and if you're really going for trajectory inference, I think there's some more specific methods really developed for that. Sometimes when you're just doing the clustering, you can pick up some patterns, and they will show up in the tree, but it's not the main goal of the algorithm.
I think very clear, very, very interesting. Well, I don't know if we still have time. You have a question, Ariel?
Yes. Is there any relationship between the number of cluster and the number of meta cluster in the flow zone?
Very good question.
Do you understand what I mean?
Yeah, and that's indeed also a very interesting question. I think it's one of the main limitations currently of the flow sum algorithm is that you need to pick these numbers by hand. So both of them, actually, you need to choose them. Typically, we go for a 10 by 10 grid in the self-organizing map, which results in 100 clusters. And then for the number of meta cluster, we typically just pick like a bit more than the number of manual populations you would expect, which is kind of arbitrary. And I know this is not like the nicest scientific explanation to give. But basically there, the idea is just that if you would take exact the same number, the chance is very big that something gets splitted that you didn't think about. And then something else will be merged that you were actually interested in. So always try to go, like if you have to choose too low or too high, you would rather go a bit too high. Because if things are split up too much, you can still easily say, okay, we will group them together, or we will add the numbers together. But splitting things up afterwards, once they're aggregated, that's very difficult. I think one important thing about this is also that it relates to your number of markers. If you have more and more markers, especially with these bigger and bigger panels, it just allows you to see more and more potential subgroups, more combinations of markers that could potentially occur. And this will also mean you need to increase both your number of clusters and your number of meta clusters. And then, for example, typically we would go for maybe a 12 by 12 or a 15 by 15 grid. So going up in the number of clusters and the number of meta clusters. So there's not an immediate correlation, like it's two separate numbers you need to pick, but typically you will increase both or decrease both depending on your panel of interest.
Okay, thank you.
Okay, maybe if we have time, one final question is, what would you recommend to deal with very rare populations with very low abundant percentage?
Yeah, if you want to detect those, I think one important remark to make is that it's not only about the percentage of cells you have, but also about how different they are from everything else in your sample. So if, I don't know, I have a sample with, I don't know what, all kind of pretty similar cells, and then I have one super small population, but they are completely different in their whole marker expression, they will still be much easier to pick up than if it's a small population that's really similar to another population, but only one marker difference. So that's why it's very hard to put an actual number on this is the percentage of cells we can still detect, because it also really depends on the actual pattern comparison to the others. If you want to do it with FlowSum, the way to go is to really just keep increasing your number of clusters, basically, until they get detailed enough that they can they can pick up this very small population. That's the only way to approach it we have in FlowSum. Like I mentioned during the talk, there are some techniques developed specifically with rare clusters in mind, and maybe these are better suited. Also of interest is that often you can do, like you could already in the lab try to enrich for your population of interest, but also in a computational setting you can still do this after you measure the sample. So maybe I have this whole sample, but I'm interested in this very small subset of T cells. Maybe I first do some pre-gating steps looking only at CD3 positive cells and starting my FlowSum analysis or whatever exploratory analysis you're using only on that subset, rather everything that I measured. And that could also be really helpful to detect these small populations.
Okay, very clear and very useful recommendation. Well, I think we are already in time, so I want to thank you so much for your generous presentation on so comprehensive and so many interesting topics. And well, that's all. And my thanks to you for the opening.
Thanks very much for giving me the opportunity to present here, and I hope it will have been interesting. Thanks so much.
Thanks so much.
Thank you very much.
Este curso de avances en citometría de flujo y citometría computacional organizado por la SAI. Voy a cambiar a inglés para presentar a Sara De Biasi. Good morning, everyone. We are starting the last part of the course organized by the Argentinian Society of Immunology. Now we will have the pleasure to introduce Sara De Biasi. Sara De Biasi is Assistant Professor of General Pathology and Immunology at the University of Modena and Reggio Emilia School of Medicine in Italy. Sara obtained her PhD in Clinical and Experimental Medicine, area of interest immunology, from the same university in 2013. From then, she has been working as postdoc in the lab of immunology directed by Professor Andrea Costariza. Most of her work was focused on the adaptive immune response during HIV infection, autoimmune diseases, and cancer. In particular, she studied the role of rare cells such as NKT cells and circulating endothelial cells in the aforementioned conditions, and more recently, her studies were dedicated to the importance of T-cell metabolism in different pathophysiological onsets. Sara is an International Society for Advancement of Cytometry, or ISAC, Marilu Ingram Scholar, and serves on several ISAC task forces. She is very much involved in teaching flow cytometry, being active part of CytoU task force and of the live education task force also. Since several years, Sara has been giving lessons on panel design in basic and advanced flow cytometry courses organized by the same task force in Europe and China. Her scientific endeavors have been presented in 75 publications in peer-reviewed international journals and more than 20 oral presentations at international meetings. Sara is also a member of the editorial board of Cytometry Part A. Today, Sara will share with us her lecture about algorithm applications in flow cytometry. Thank you, Sara, for being with us and share your knowledge and expertise with Argentinian immunologists.
Thank you very much for the invitation. I start to share my screen and the presentation is here. So good morning to everyone. As Florence said, that today we will talk about clustering methods impact in immunology research. If you have any question, please contact me at sara.debiasi@unimore.it. This is my email, and I reply if you have any question about this presentation of today. I'm a biotechnologist, and during the last year, I tried to use this kind of clustering methods to analyze my immunology research. So this is the overview of today. So we will talk why we should apply dimensionality reduction in immunological studies, what we have to do before dimensionality reduction, so the importance of generating good data, because out of bad data you won't have good data. And I will try to explain to you 3 different examples on about the clustering methods in 3 different onsets. So HIV infection, cancer. So I will show you data obtained in melanoma patient and renal carcinoma patient treated with anti-PD-1, so immune checkpoint inhibitors. And finally, I will close my talk with the SARS-CoV-2 infection and COVID-19, with a focus on T, B, and monocyte immunophenotype and function. So we have to start, and I think that it's not what I think, but it is the truth. The oldest single cell technology, the oldest single cell methods is represented by flow cytometry. Flow cytometry, for instance, is able to analyze the expression of different proteins, up to 40 proteins by now, on a single cell level. And single cell analysis is also the study of genomics, transcriptomics, proteomics, metabolomics at a single cell level. And it is very important because it allows us to discover heterogeneity in the immune system and immune cell population, because it is very different to analyze a lot of markers in a bulk population, and it is very different to analyze a lot of markers at a single cell level. And single cell technology is very interesting because it can allow us to discover new cell population not seen when studying bulk population of the cells. So imagine that you are running a flow cytometry panel with 20 colors in high demand, and you will have a lot of dot plot to analyze, because, for example, every plot is bidimensional. So you will have 2 to 20 population to analyze, and of course the analysis will be driven by the expert that is doing the analysis. So maybe you can lose heterogeneity because you don't know an existence of a population in your clusters. So why we perform dimensionality reduction? First of all, to simplify complexity, so it becomes easier to work with your data, and you have to renumber the numbers of features of markers. You can remove redundancies in the data. You can identify the most relevant information, because you will be able to find and filter the noise. So you have to clean your data before doing unsupervised analysis. You can reduce computational time for downstream procedure. You can facilitate clustering, since some algorithms struggle with too many dimensions. And at the end, you can also visualize your data by using different methods, and you can select which one you prefer. So there are different ways to analyze analysis. Any way depends on the different computational pipelines. And here it is shown an example obtained with published data, in which you can see naive CD40 cell, CD14 monocyte, memory CD40 cell, B cell, cytotoxic CD8, other monocytes expressing CD16, NK, and dendritic cell and platelet. And these are all different data visualization methods. So for example, PCA, the principal component analysis, or TSNE or EUMAP. And you can see here that on the basis of the graph, you are looking, you will see the same results. So different kinds of population clusterized in different parts of the plot. And you can see here, for example, with Disney or with UMAP, that the population are really separated by each other. So by selecting a computational tool, you can analyze your data and you can visualize your data and see which kind of population are represented in each data set. The Shapiro-Stephens law of flow cytometry is that no data analysis technique can make good data out of bad data. So it's what I was saying to you before. So if you want to have a good analysis, you have to clear your data, you have to perform a good acquisition at your flow cytometry, a good staining to your cells, and of course you have to take into your minds a lot of rules of flow cytometry in order to generate a really good data, because at the end, even if you are cleaning your data, if your data are not stained well, you will not obtain good results. So in 2010, Enrico Lugli, Mario Roeder, and Andrea Castorizza published this paper in Cytometry Part, in Cytometry Part A. And in 2010, there was not a lot of unsupervised method of analysis, because mostly we were performing manual gating based on sequential gating. The mode of visualization was histograms or dot plots. So we were analyzing few markers, and it was everything operator dependent. But nowadays, but also in 2010, PCA-based method came out, and this kind of method were really important because they were operator independent, and it allowed to you to perform an unknown population analysis. So this is the main rules of gating strategy you have to follow in order to clean your data and to find the population of interest. So first of all, you have to perform the debris exclusion. So you have to avoid from your analysis all the debris. You have to perform the singles and double discrimination by using, for example, one dot plot of forward scatter area versus the height, and also you can use also the width. You have to exclude cell deaths if you are working with, for example, a thaw PDNC. So you have to use a viability dye. You can use a dump channel in which you can put all the cell type of interest versus the relevant. So a channel in which you put all the unwanted marker in your analysis. And of course, you have to exclude events during fluidic issue. So for example, you have to use the time gates, and then you have to remove the fluorochrome aggregates by using the keeper gate, because for sometimes during your staining, some kind of fluorochrome aggregates could form in your cocktail antibody mix, and you can recognize this kind of fluorochrome aggregate because they have a particular pattern. They are super bright, yeah, they are very particular, and they are at the edge of your dot plot. In 2016, this review came out in Nature Reviews Immunology. It was written by Ivan Sayes and Sophie Van Gassen, and it was very useful for me because it was a complete overview to all self-organizing techniques to visualize data. So starting from the oldest, which is Spade, that was published, I think, in 2011, 2012, to other kind of visualization and clustering methods. So if you want to have an idea of all the, I mean, this was 2016, but I think that if you want to have an idea on how this kind of methods work, you can download this review. Today, I would like to talk about Phenograph because we applied Phenograph in two different papers we published recently, and the goal of Phenograph is the automated partitioning of high-dimensional single-cell data in subpopulation. And Phenograph is really important because it can map the nearest neighbors. Phenograph is able to clusterize your data on the basis of three orders of relationship. So the first order of relationship is that Phenograph find the key nearest neighbors for each cell using Euclidean distance. The second order relationship is that cells with the shared neighbors should be placed near one to another. And third, it identifies communities by Leuven methods that measure the density of edge inside the communities to the edge outside the communities. Of course, Phenograph is characterized by advantages and limitations. Advantages is that it can give you the opportunity for population discovery. It can resolve subpopulation as rare as 1 in 2,000 cells. It is very robust to cluster shape. In fact, it does not need to be spherical. But it has also some kind of limitations. So, for example, is the user that specifies the number of neighbors, and the ideal cluster number or biologically relevant cluster number is largely unconstrained. So you have to try different kind of K to see which one is the best representative for your experiment. And in 2020, we published this editorial review with data analyzed with different methods of unsupervised analysis. So, for example, here in the left, you can see the heat map coming from analysis performed with Phenograph in CD4 and CD8 subset in patients during heart. And thanks to Phenograph, we were able to identify different subpopulation of T cells among CD4 and within CD8. So we were able to identify different clusters of central memory, one cluster of naive T cell, 2 clusters of central memory, and 1 cluster to effector memory, and 1 cluster of terminally differentiated effector memory, and 1 activated or exhausted transitional effector memory. And all of this was possible by using a total of a multicolor panel of 16 colors, in which we clean all the unwanted events, such as debris, and as I told you before, dead cells. Here in the right, we have another kind of examples of cytokine production by different types of CD4 and CD8. And this kind of clustering was performed by using flowsome and represented by UMAP. And also here, we can see that after stimulation, cells from HIV patient were able to produce different kinds of cells, such as interferon gamma, TNF-alpha, and IL-17. And as you can see here, mostly the cytokine producing cell was effector memory, central memory, and transitional effector memory T cells, and were clusterized here in this kind of islet. And here we can see that, for example, we have an effector memory population characterized by the production of interferon gamma and TNF-alpha, so we call it TH1 population. And the same, we were able to see also the same cytokine production within the CD8 cluster. So also here, thanks to 2 different markers such as CD45RA and CCR7, we were able to identify different subpopulation of this cell, and in particular, we were able to find population that were able to produce cytokines after in vitro stimulation. And most of these cells were characterized by the presence, by the expression of the absence of expression of CD45RA and CCR7. So they were effector memory T cells. The first example I would like to share with you is this kind of paper we published in the Journal of Infectious Diseases in the middle of 2020, in which we publish data regarding a particular cohort of patients, of HIV patients receiving an orthotropic liver transplantation. So the aim of this work was to understand the interaction between the immunosuppression caused by the therapy and that caused by HIV infection per se, because we understood that this is crucial to understand whether a successful transplanted patient can respond or not to pathogens. And of course, there was a big gap in the literature because most of the immunological studies on the effective immunosuppressive drugs on T cell have been performed in animal model or healthy volunteers. So there was no data in the literature regarding immunosuppressed patient with HIV. And for us, it was very important because we wanted to understand why sometimes in this kind of patient there is the graft rejection or the upcoming viral infection. So we study a total cohort of 180 subjects. There were 27 subjects per each group, and we have 4 different groups. So we have patient healthy donors that we call controlled, HIV positive infection but without transplantation, people with transplant without HIV, and of course people with HIV and that received transplantation. And these were our cohort of interest. Patients were matched for age, were matched for sex, and they were not matched for ACV because a patient with, of course, that receive a transplant and with HIV had higher percentage of people with HCV. These are the characteristics, the patient clinical characteristics of the HIV infected patient. So we have here all the CD4 count. They were similar for CD4 count, for CD8 count, for the ratio, for the nadir, CD4 nadir, and there were differences in the therapy. There were kind of differences in the therapy because some of them were treated with Maraviroc or with integrase inhibitor, but the time of art was completely similar. And here we have the clinical characteristic of a transplanted patient. So as you can see here, they were all similar for the type of immunosuppression, for the for the time between the transplant and the sampling of the samples, and they were different, of course, for CMV reactivation and for alt indication. This is the experimental workflow. So we take fresh blood, we process it into single cell suspension by performing FICOL and isolating PBSC, and we perform a 16 color for cytometry staining by using Duraclone and adding other from Beckman-Coulter, and by adding other kind of antibody previously titrated. We were able to identify CD4 and CD8 cells. We perform Phenograph clustering, and we visualize all the clustering by t-SNE. And then we identify the Phenograph cluster by performing an integrated medium fluorescence intensity, and we analyze also the frequency of this cluster. This is a deep T-cell characterization. As you can see here, there is an MPRN matrix, very useful to check all the samples and very useful to check the compensation. So first of all, you have to check the compensation if in different days, if you want to be reproducible and apply this kind of unsupervised cluster of analysis. So in our panel, we were able to identify CD4 and CD8, different type of subpopulation of T cells, such as naive central memory, effector memory, terminally differentiated effector memory, together with the expression of PD1 and CD57. And of course, we were able to analyze putative Tregs on the basis of the expression of CD25 and lack of expression of CD127. And we were also able to analyze activated T cell, but also different subpopulation of monocytes on the basis of the expression of CD14 and CD16. Another thing that I would like to underline is the machine performance across the independent experiment, because of course you have to be very reproducible. So you have to understand if your machine is working well and it is reproducible day by day. So if you are running different samples per day, you have to check the machine, but also to have one control that you can run every day in order to see if the control is the same for every day, in order to avoid the batch effect analysis. And also you have to check also that you have to have similar distribution of compensated data between independent experiment. And I suggest you to perform a matrix overlay of antigen expression among the different runs. If you have checked all of these points, maybe you can clear your data and you can perform an unsupervised clustering analysis. So first of all, we didn't find any differences among the CD3 T cells, or we found that patients with and without HAV were characterized by lower level of CD4 and higher level of CD8. So the cytotoxic compartment was higher in terms of percentage. And here you can see the similarly different distribution of CD4 T cell cluster amongst the different groups. And here you can see, for example, the overlays of T cell distribution of different patients on Disney plot. So different color in this case represent different patient, and you can see here that different area of the plot are occupied by, for example, different patients. These are the cluster we found in the distribution of on Phenograph, and in this graph, different color represent different clusters. And here, for example, we found all the T cells population among CD4 T cells. As you can see here, we were able to analyze 7 clusters central memory, 4 clusters of transitional effector memory expressing PD1, naive T cell, effector memory expressing CD57, so cells that are going to reproductive senescence, effector memory T cells, and also Tregs. And we perform analysis in differential analysis to understand if the percentage of this cluster was different between patients and the different kind of patient and control. And we reported the stars that is represented by P less than 0.05. And in particular, we wanted to analyze the percentage of this cluster. And what we find out is that, for example, this kind of cluster, so cluster 19, was really high represented in HIV negative patient receiving orthotropic liver transplantation. And this kind of population was a population of effector memory T cell expressing PD1. In order to confirm this data, we also went through manual gating. So putting a gate on CD4 positive, CD3 positive, and CD45 positive cell, we were able to identify CD27 and CD28 cells positive, and then we analyzed those positive for CD106, 127, and in this population we were able to analyze those expressing PD1. And we characterize also this population for other markers, so they were all CCR7 negative, CD25 negative, and HLA-DR negative, and we confirm that we were able to find the same differences even with the manual gating. We performed the same reason for CD8 T cell cluster. So also here we were able to find different cluster of effector memory T cell, those more differentiated and activated, and those more indifferentiated, such as naive T cell. And what we find out here is that in patients that were HIV positive and receiving transplantation, if compared to patients with those receiving transplantation, we found here that this kind of cluster were more represented. And we confirm also this by performing the analysis, the differential analysis between the different frequencies. We were able to analyze also cytokine production by stimulating cells in vitro with anti-CD3 and anti-CD28. And what we found out is that CD4 and CD8 T cells of patients receiving a transplantation were characterized by a higher level of cell that are able to produce interferon gamma and TNF alpha, and also IL-17 within CD4 T cell subset. About polyfunctionality, we found that this kind of cell were characterized by Th1 properties in CD4 T cells and also CD8 T cells. So concluding this kind of first part, we can say that we found differences in patients receiving autotrophic liver transplantation without HIV, that they are characterized by a more exhausted T cell phenotype if compared to HIV positive and OLT positive patients. And this kind of results were kind of unexpected, but a possible explanation could be that patients that without HIV and receiving autotrophic liver transplantation have been living for several years under an immunological disruption that is different compared to what that of HIV positive patient and other factors. So for example, IL-10, TGF-beta, and TG load and persistence. So we can explain that maybe this kind of patient underwent to a stronger response to allogenic MHC molecules, leading to an exhaustion of functional reserve. I just want to go to the cancer section. We published this paper in the first month of 2020. This is a review, and we analyze the different single cell approaches to profile the response to immune checkpoint inhibitors. So here we took into consideration all the advantages and disadvantages of the cutting-edge single cell technology to profile cancer immunity. And as you can see here, flow cytometry is the flow cytometry, mass cytometry, and flow cytometry in general are the first considered, because they are the oldest one. And here I report a path to biomarker discovery. So this is a computational workflow for single cell experiment. So first of all, if you want to undergo to a canonical single cell experiment, you have to take into consideration all of these kind of steps. So for example, you have to study the experimental plan, you have to set up and generate the data, of course, try everything, standardize everything. You have to perform a very good data pre-processing, so you have to perform quality control, you have to clean your data, you have to check for technical issue and batch error, and then you have to perform data analysis, and in particular data visualization that is based on 3 different steps. So for example, cell type identification, differential analysis, and data integration. So you can find everything in this kind of review. And I want to share with you, with all of you, data we published. We recently published, the paper is submitted to Nature Communication, and we hope to receive news soon about this kind of paper we published in studying PBMC isolated from metastatic melanoma patients treated with immune checkpoint inhibitors. And what we find out is that circulating mucosal associated invariant T cell can identify patients responded to anti-PD-1 therapy. So this is a little bit of background. So CD8 is able to drive adaptive immune responses against several types of human malignancies, but in particular, if it can drive adaptive immune response, all of these high mutational burden and new antigen load present in the tumor can drive to T cell exhaustion, but this kind of immune checkpoint therapy are able to reinvigorate T cells in several cancer settings. But of course, not all of patients respond to this therapy, and the mechanism and the molecular mechanism behind this kind of response and not response are still not clear. So there is a pressing need to deeply interrogate immune cells, either in the tumor microenvironment and in blood, has led to investigators to integrate data obtained from traditional approaches with those obtained with new, more advanced single-cell technology capable to define characteristics of the immune cell at an unprecedented degree of resolution. So we study a total of 28 patients. We were able to characterize 11 not responding patients and 17 responding patients. They were very similar for mean age, sex, and the stage of disease. None of them was treated with previous therapies, so they started therapies with anti-PD-1, nivolumab or pembrolizumab. And this is our experimental workflow on PBMC isolated from patients with metastatic melanoma. So also here we have investigated 3 different time points. So time 0, which is before therapy, time 1, which is after 1 cycle of therapy, and time 2, which is after 2 cycles of therapy. Of course, we took blood, we analyzed PBMC, we performed different kind of clustering, and then we were able also to sort CD3 positive, CD8 positive T cells, and we performed the analysis of transcriptome by single cell RNA sequencing. And of course, we correlate all of this data with the correlation with data with the clinical outcome. So the disaggregating strategy again used to identify CD8 positive T cells. So also here we clean all the data, we identify CD3 positive, we put a keeper gate in order to remove this kind of aggregates and unwanted population. We clean for physical parameters. We put a gate on all the fluorochrome aggregate, and then we were able to identify CD8 positive T cells. And in this population, we perform Phenograph clustering, and this kind of high-dimensional single-cell analysis of CD8 positive T cell was able to identify higher percentage of activated effector memory T cell in responders. So as you can see here, cluster 16 were characterized by higher expression of Ki67, ICOS, CD95, HLA-DR, CD71, CD98, higher level of expression of granulizing and CXCR6 and CD38. So this kind of expression, this kind of heat map was able to identify effector memory T cells proliferating because characterized by HLA-DR and Ki67. And this kind of the most important things we found here is that this kind of cluster were representative more in responders at time 0, and then this kind of difference was even maintained after the first cycle of therapy and after the second time of therapy. We wanted to have a major overview. We wanted to go more in deep in the knowledge of CD8 T cells coming from this kind of patients. So we sort the CD8 T cell and we perform single cell analysis. And what we found here is that in this kind of human, all the population we found are represented. So we found a population of naive T cell, a population of cytotoxic, terminally differentiated effector memory T cell, transitional memory T cells, 2 population of naive T cell recently activated, once expressing alpha-3 and the other expressing FOS and June, activated effector memory T cell, and we found also a population of mucosal associated invariant T cells, which was not characterized by our flow cytometry analysis, because in the panel TCR 7.2 and CD161 was lacking. So we perform also pseudotime and different kind of analysis. And what we can see here is that the differentiation pathway starts from here. So you can see here pseudotime starting with naive, and then we have the 2 population of recently activated naive T cells, effect transitional memory T cells, cytotoxic EMBRA, and here we can see a branch which correspond to mucosal associated invariant T cell, which mainly are characterized by effector memory T cells able to produce pro-inflammatory cytokines, but also they can exert also pro-cytotoxic properties. And here, all the genes, all the important genes we found that characterize different population. So, for example, this kind of mite cells are characterized by the expression of KLRB1, by SLAC4A, MAF, which is an exhaustion marker, and CXCR4, which is a chemokine receptor able to induce the migration of these mite cells. All the other population were characterized by the expression or the lack of expression of memory and naive genes. So also by single cell RNA sequencing, we confirmed that this kind of cluster 16 we found on Phenograph was also present by performing this kind of single cell RNA-seq analysis. So for example, here we see that they are higher after 2 cycles of therapy. And this kind of population were characterized in responding patients by a higher level of activation of, by level of expression of activating genes such as FOS, DUSP, and also HLA-C, and higher level of granulizing and granzyme H. But what we found really interesting is that we found that the proportion of mucosal associated invariant T cell in percentage was higher from time 0 and this kind, so before therapy, and this difference was maintained also after 1 cycle and 2 cycle of therapy. Not only the proportion was different, but also from a gene expression profile, this kind of cells in responder were characterized by higher level of activating markers, such as, for example, FOS, Jun again, and DUSP, and the higher level of granzyme K and CXCR4. In order to understand better understand what was happening in the mite, we reclustered the mite subset, and this kind of reclustering was able to identify 2 different clusters inside the mite cell population, such as the not activated mite represented here in salmon, and the activated mite represented here in blue. And you can see here that not activated and activated mite were characterized by differential expression of activating gene, and also such as CD69 and FOS, but also other kind of genes such as TNFA AIP3. And what we find out is that activated mite cells are more abundant in responders if compared to non-responders. And you can see here by this kind of human representation, you can see that the responder patients are characterized by higher level of activated mite here in blue, and if compared to not responding. And here you can see the graph. So this kind of activated mite was present even before therapy, and this kind of difference was maintained after the therapy. We wanted to confirm this data by using flow cytometry, so we ran a flow cytometry panel, an 18 parameters flow cytometry panel. We were able to identify this kind of mite cells on the basis of the expression of CD8 and the expression of TCR7.2, which is the characteristic TCR receptor of these cells, together with higher level of expression of CD161. And what we find out here is that we found higher level of mite cells in responder if compared to not responder patient. And the other data we confirm is that during therapies in responder patient, there was an activation, an increase of level of expression of CXCR4. So we hypothesized that this kind of cell were able to migrate into the metastasis and primary tumor site. Unfortunately, we couldn't have skin biopsies from patients during therapy. So Dr. Domenico Tartaro performed a GSA analysis to analyze if this kind of cell were able to migrate into lymph nodes and tumor. And what we find out is that this kind of cell, so mite cell characterized by the expression of KLRB1, CD69 and CXCR4 was able to go into the tumor, and we found a really high overlapping p-value between our mite signature and the mite signature of published data. So we suppose that these kind of cells are able to undergo into the metastasis. Finally, we analyze the mite polyfunctionality after in vitro stimulation, and what we find out is that this kind of cells after stimulation with IL-12 and IL-18 were characterized by higher level of production of granzyme B and interferon gamma. Regarding polyfunctionality, what we can say is that the population, the cells that are able to produce granzyme B and TNF alpha was higher in responder if compared to not responder. And if we analyze the only production of granzyme B, we can see here that responder patients are characterized by higher level of granzyme B production. Finally, we correlate all of this data with clinical data. And what we found is that the level of mite of cells before therapy could identify responder patients. So we took the median level of mite cell that were 1.7 out of 1.7% of CD8 positive T cell, and we stratified the patient on the basis of response to therapy. And what we found here is that this kind of value was able to identify patients who will respond to therapy. So in order to wrap up this second part of the talk, we can say that patients who respond to immune checkpoint inhibitors are characterized by different composition of T cells the population compared to those who do not respond, that are detectable before the initiation of therapy. And we focus our attention on mucosal associated invariant T cell because we found a lot of differences in this population within responders and not responding patients. And we suppose that this kind of mite cells could be a predictive marker of successful therapy. And this is quite interesting because very recently a population of MHC class 1 related molecule restricted T cells belonging to the family of mite, and this kind of cells are called MRR1 T cell, has been described as a rare population able to respond to a variety of tumor cells of different tissue origin, but not to microbial antigens. So further studies are needed in order to understand if this kind of cells can be represented by this kind of population. The second example I would like to bring to you is another study we perform on metastatic renal cancer carcinoma patients treated with immune checkpoint inhibitors. Renal cancer carcinoma account for 5 and 3% of all malignancies, and 15% of these are metastatic disease at the diagnosis. And this kind of metastatic renal cell carcinoma has a still approved prognosis. And even if in 2015 there was an appreciable increase of life expectancy due to the introduction of immune checkpoint inhibitors such as anti-CTLA-4, anti-PD-1, and anti-PD-L1, a lot of patients do not respond to therapy. And of course, also here, the reason at the basis of the lack of response are still largely unknown. So it is also very complicated to have biopsies of these patients. So we thought that by analyzing PBNC in this patient treated with anti-PD-1 could help us to find some kind of biomarker to identify patients who will respond or not to therapy. In order to analyze this kind of data, we use a new interpretable machine learning methods for single cell data that can reveal correlates of clinical response to cancer immunotherapy. This kind of method is really new. It's still published still present on bioRxiv, but it was presented at Virtual Cyto last August. And it is completely new, but really robust because it is based on annotation forest. And this kind of method is able to perform unsupervised analysis on the basis of the marker you are using, and in particular is able to decide which kind of markers are really useful for your analysis or not. So we applied this kind of methods to 19 patients with renal cell carcinoma. Here are all the clinical characteristics. This paper is unpublished. We are still preparing the draft, so this kind of data are really, really new. And these are the markers used for the clustering and protein abundances expression. So we use 28 color panels, but all of the cluster was really important to identify the population of interest. And thanks to this marker, so marker related to the differentiation status and also to the function profile, so T-beta expression, we were able to identify different 59 cluster actually, that was representing all the spectrum of CD8 T cells. So we found 4 clusters of naive T cell, different 2 clusters of T cells and memory, different cluster of central memory, different cluster of terminal transitional memory, different cluster of effector memory, and a lot of cluster of terminally differentiated effector memory. And here you can see the distribution of all these clusters within CD8 T cells. And what we find out is that this kind of patient treated with anti-PD-1 display an increase of effector memory compartment and a decrease of T cells and memory percentage after therapy. And of course, this kind of data, we have to finish the experiment, and there are a lot of investigation going on. But of course, what I can say is that, for instance, for sure, this kind of therapy is able to induce a redistribution of T cell population. And last, I would like to focus my attention on the COVID-19, so SARS-CoV-2 infection. We work a lot in the first wave period, so in the first, because we were the second place in the world where COVID arrived. First was China, and then it arrived in Italy in March and last of February, first days of March. And we were able to study blood isolated from this patient, and we focus our attention on TB and monocyte immunophenotype and function. And this kind of data will be discussed by Professor Andrea Costarizza in the next day during a seminar dedicated only on COVID-19. What we were able to characterize is that this kind of patient were characterized by cytokine storm, in which the body of the patient with the SARS-CoV-2 infection perform a desperate attempt to respond to viral infection in a very unorganized way. We found a complete subset of monocyte that is completely dysregulated from a metabolic point of view, and T and B cell were completely altered because T cell were characterized by higher activation in terms of CD38, HLA-DR, and PD-1 and CD57 expression, and B cells we find really high level or percentages of plasmablast, and in particular we find a population of senescence B cell, and we did not find any difference in B cell proliferation. And here, for example, we published this paper a few months ago about monocyte phenotype in COVID-19 patient. We perform a very easy flow cytometric panel involving the study of CD14, CD16, PD1, TIM3, PDL1, CCR2, and CD38, and CXCR3. And we were able to characterize by flowsome and represented by UMAP 4 different clusters of monocytes. So first of all, we identify classical monocytes on the basis of the expression of CD14 and CD16, and 2 different subpopulations of intermediate monocyte on the basis of the expression of CD14, CD16, but also CCR2, which is a receptor that is able to induce migration of monocyte. And then we identify non-classical monocyte on the basis of expression of CD16 and CD14. And what we find out is that patients with COVID infection, with COVID, were characterized by higher level of intermediate T cell and lower level of and higher level of classical monocyte if compared to healthy donor matched for sex and age. Another example is this one. We applied the same method to perform clustering among CD4 T cell from COVID-19 patient. And also here, what we find out is that with the marker we selected, we were able to cover all the spectrum of CD4 T cells, starting from naive, arriving to T cell zone, which is characterized here. For example, here we find naive Treg, central memory Treg, and effector memory Treg. And here we were able to identify effector memory Treg and those expressing, and those more senescence expressing CD57 and PD1. And we found here also differences in terms of percentage of naive Treg, effector memory expressing PD1, central memory Treg, activated central memory PD1. The same methods were performed among CD8 T cells. So also here UMAP was used to visualize this data, and FlowSum was used for clustering. And what we found out here is that we found a lot of that the COVID patients were characterized by higher level of terminally differentiated effector memory T cell that express CD38, so they are activated, expressing senescence marker, expresses PD1, lower level of central memory, and higher level of effector memory expressing CD57. And finally, I would like to show you data we publish about B cells in COVID-19 patient, and also here the same methods were applied. So here we were able to identify different cluster of B cells, spanning from naive B cells arriving to unswitched CD24 B cells, which can represent a cluster of senescent B cell. And what we find out is that all the subpopulation of B cell are completely dysregulated in COVID-19 patient, and in particular, we found really high level of plasmablast if compared to healthy donor. So I like to wrap up and give you some kind of future direction. So I hope to have convinced you that the application of single cell technology has generated and will generate in the upcoming years an explosion of new data with a clear impact in the translational clinical research, thanks to the identification of possible biomarkers. And it is likely that the huge amount of information will also revolutionize the fields of basic of research immunology and cancer biology. So we should do a big effort, because we have to make all the data, including the raw ones, available to the scientific community, and to create the rings for data extraction. The information gathered from these technologies will add novel hallmarks of response to immune therapy that could be integrated in the routine clinical management. Nonetheless, this kind of research, the root of the discovery of new biomarkers is still bumpy, because, of course, we have to pay a lot of attention in how we handle all the cells during pre-processing, and an adequate data analysis with potential bioinformatic tools are critical factor to preserve the native biological profile that will ensure meaningful conclusion. And finally, investigating the role of exhaustion and or dysfunction and translating this knowledge to clinical practice can be considered main challenges in the battle against cancer and viral infection. And having said that, I would like to thank you for your attention.
Okay, thank you very much for your excellent talk. Amazing work you do, Sarah.
Thank you very much. It's a big team working on this.
Yes, I can imagine.
On this data.
I can imagine, but it's amazing. It's amazing the stuff you do, and all the work and the information that you get. It's amazing. It's amazing. Here in Argentina, we don't have such kind of technology. So it's amazing that you can bring that to us and show that kind of work, which is amazing. So we still don't have any questions from the audience, but I have I can start asking you about the Phenograph. You said that you can see one cell from 2000 in Phenograph. And how many markers do you use to identify 1 out 2000 cells in Phenograph? Because could you explain that to us?
Yeah, this is a very good question, because of course this kind of analysis should be tested, of course, every time you perform it. So this kind of Phenograph, this kind of analysis has been published, saying that you can find 1 cells out of 2000 cells. But of course, you have, as you said, you have to clarify how many markers are you using, and in particular, you have to test the K, because the K is very important, because the K can establish how many cells are around other cells. So on the basis of the K, you can discriminate different cells or the same cells. So I'm sorry, but I don't have a good answer for this, because you have to test your system every time with Phenograph. And for this reason, is a little bit, I mean, it's good, because it is very good. You can also avoid batch effect, because then at the end you will find your marker in the analysis if you have to remove before it. But of course, you have to test everything and be sure of what you are doing.
Thank you. And regarding, as you mentioned, batch effect, and how do you correct batch effect with your data? Do you use any normalization? When you see a batch effect in your data, what do you do?
I take the files and I send the files to our bioinformatics. No, I'm joking. I'm sorry about that. No, I mean, if we see this kind of batch effect, one thing you can do is that you have to normalize. You can use 2 different tools. You can use Cytonorm. You can try to use Cytonorm that Sophie Van Gassen published, I think, last year or 2 years ago, and try to normalize and try to rescue the value affected by batch effect. Or otherwise, if the batch effect cannot be removed, you have to avoid to use the marker, for example, affected by batch effect. But batch effect is very annoying. I mean, it could be, I mean, you can lose a lot of data because of batch effects. So that's why I recommend every time before starting every kind of experiment to check if the instrument is performing in the same way like the day before, and the staining procedures should be really the same. And I suggest not to change the antibodies used in your flow cytometry panel if you have to perform this kind of analysis, because of course this kind of batch effect are really tricky to remove, are not easy, and they can make you lose a lot of data.
Great.
Yeah.
So you have to think a lot before starting your experiment.
Yeah, and you have to test it several times.
Yeah. Great. I would like to ask you about COVID-19, because you showed a lot of data showing the people who has the disease during the disease. But what happened with convalescent people? Do you check with convalescent, because it has to do with vaccine development? So I would like to ask you about your results on convalescent persons.
We start calling back all the patients that was convalescent from COVID-19, and we are collecting the blood. So we are starting our experiment within within really few weeks. And what we would like to do is that to reperform the same experiment in the second in this kind of second time points. What we expect is that patients that fully recover increase their level of leukocytes, so these patients are not a leukopenic anymore, and also we expect that all the activation marker we found probably they went down. But of course we have still to try this kind of experiment to see what is happening. We have data regarding the antigen specific stimulation, so we tested the amount of cells that were able to respond to different type of antigenic stimulation, and we saw kind of differences between mild and severe patients. But of course we have to finish the work. So I mean, we have data but not conclusive one by now.
Great. Did you analyze antibodies, neutralizing antibodies, and the relationship with the T cell compartment or B cell compartment?
We are doing that. Yeah, we are collecting plasma and the cells, and of course we will do that for sure.
Lots of work.
Yeah, yeah, a lot.
Lots of work.
and still doesn't seem to finish.
Yeah, you're right.
Here comes a question from the audience. It says, is it okay to regroup different clusters into biologically relevant groups? For example, C1, C3, and C5 are together CD8 T central memory. What do you think of the personal bias when grouping them?
Yeah, this is a very good question, because of course sometimes we want to group all the different clusters in order to, because when we see the heat map, maybe we can recognize that the patterns are not so different. So we tend to cluster them together. What can I say is that you have to pay a lot of attention when you are doing this kind of stuff, because of course you can lose the heterogeneity you are looking for. So my suggestion is that if you identify different clusters, for example, of central memory or effector memory T cells, which are similar on the basis of the integrated MFI or protein expression, my suggestion is to have a look at the percentage of the cluster and the reason to that. So if the cluster is very low represented, maybe is the same of another cluster, and maybe you can put together, and you have to take into consideration also where this cluster in the UMAP map are. So for especially if they are spatially divided or if they are spatially similar. So we perform some kind of reclustering. We add a different cluster together, but we perform a lot of thinking. We do a lot of thinking about that, because of course this kind of methods allows you to find heterogeneity, and if you recluster them together, maybe you can lose this kind of heterogeneity.
Congratulations. Sorry, congratulations for your work and presentation. I would like to make a question. What do you think Phenograph or flow cytometry is better for discover a new population?
This is a very good question. I think that sometimes you don't have an answer for that, because the computational methods is different. But I think that if your data are really strong, and if the differences are really strong, all the methods should give you the same, more or less the same results. I think the Phenograph is very, very fast, and it allows you to save a lot of time during the analysis, while Phenograph is really quite more tricky, because of course you have to select the K and rerun everything, and then to perform the integrated median fluorescence intensity map, you have to perform a manual gating strategy. So I think that FlowSum with the UMAP representation is faster and can gives you a better representation of all the heterogeneity. Phenograph is quite more long, but at the end the results are more or less the same. If the K is selected in a good way, they are more or less the same.
Very clear. Thank you very much.
Thank you.
Can you tell us, Sarah, what algorithm did you use for trajectory inference in your study?
Yes, we use pseudotime, monocall. We use monocall to do this kind of analysis. Yeah, Dr. Domenico Tartaro performed the analysis, and he used monocall to perform the analysis. So pseudotime and monocall.
And have you conducted that kind of studies that was in single cell RNA data?
Yes,
you
yes.
conducted that in flow cytometry data as well?
We perform this kind of study into two different parts. So the first, the discovery one, was performed by flow cytometry. So we apply a 28 color panel to identify different subpopulation of T cells, and then we sorted CD8 T cells and we perform single cell RNA-seq, and we confirm some kind of data with flow cytometry. So we performed two different experiments in the same way, at the same time, single cell and flow cytometry, and then we use one technique or the other to confirm what we find with different techniques.
I was wondering about that integration between both platforms, how the analysis has a tendency to come together, the single cell and the...
Okay. I think that the scripts are completely different, because for single cell RNA-seq, we use Serot, all the pipeline published with Serot, so Satija Lab, and with flow cytometry, we use Phenograph. So I don't know if there is a way to combine flow cytometry and single cell data together, because these kind of metrics are quite different. But I think I can ask, because I'm not the expert in this kind of computational stuff. I can read it, but I cannot do that. So I can ask, but I don't think there is a way to put together flow cytometry data and single cell RNA data in the same metrics, but I can ask.
I have one last question. In your experience, which markers are best suited for computational analysis? Markers of memory effector phenotypes?
It depends on your biological question. I think that if once you identify which one of biological question you want to answer, I think you have to perform to build the panel and identify the most relevant markers. Of course, if you are using, for example, thaw PBNC, you have to pay attention to avoid markers that can be downregulated during the freezing and thaw. And of course, you have to put into the panel some kind of viability marker in order to identify and remove that cells. But I think that at the end, if you want to analyze the spectrum of T cells, a marker of differentiation, activation, markers of intracellular marker, for example, for proliferation, or Tregs identification can be used as well. And they are very clear. If the reagent is very clear, so if the antibody is a good antibody, you can use it even if the expression is really low. But if the separation is very good, everything can be performed.
Great. Well, we don't have any questions from the audience, so we thank you very, very much for your presentation, and it was really great for us to listen to your experience, and it's we really thank you for sharing with us, with all the Argentinian immunologists, your experience. Thank you very much, Sarah.
I thank you for the invitation, and I thank you for the opportunity you gave me to share my research with you. So thank you very much.
Thank you very much, Sarah, for your excellent presentation, your terrific result. Thank you very much. We appreciate very much that you participate in this course. Thank you.
Thank you.
Okay, well, now we will switch to Spanish, and quiero agradecerles a todos la presencia aquí. Esperen un segundito, no se desconecten, que Guillermo ahora va a mostrarles bueno, algunos enlaces que pueden seguir para obtener información de repositorios. Así que les pedimos que permanezcan unos segundos para luego hacer el cierre del curso.
Bueno, creo que están ahí visibles. ¿Se ven?
Sí, sí, se ve perfecto.
Bueno, algunos ya se mencionaron, el Flow Repository, el GitHub para los programas, Cytobank, y el acceso a los laboratorios. Cualquier cosa, cada uno de los bueno, los laboratorios ponen a disposición no solo las datasets, sino también los scripts que desarrollan y se pueden probar. Creo que lo que mostró Carla el otro día ejemplifica muy bien cómo se puede acceder a los datos y se puede reproducir lo que está publicado en excelentes revistas, y es una muy buena forma de entrar en el tema, hacer ese trabajo de acceder y reproducir esos análisis. Particularmente los datasets de OMIPS creo que van a ser muy interesantes. Así que, bueno, eso es todo, creo, ¿no, Analia?
Sí, sí, perfecto. Bueno, Florencia, si puedes aparecer, y Guillermo, entonces dejamos de compartir la pantalla, así le damos cierre al curso. Pero queremos, desde la Sociedad Argentina de Inmunología, les queremos agradecer a todos ustedes por la asistencia y, bueno, en especial a todos los disertantes que prepararon estas excelentes presentaciones, y a los coordinadores del curso, Guillermo Blanco, Ariel Villordo, Florencia Quiroga y Fernando Chirdo, que hicieron una excelente coordinación de todas las presentaciones y contribuyeron a la organización de este curso, que creemos que va a ser de suma utilidad, no solo para los miembros de la Sociedad Argentina de Inmunología, sino también para todos aquellos que hacen citometría. Queremos recordarle también que existe el Sistema Nacional de Citometría y que a través del sistema pueden tener acceso a distintos equipos que están a lo largo del país y que pueden acceder a tecnologías que quizás en sus propias instituciones no dispongan. Así que, bueno, visiten la página del Sistema Nacional para ver a qué equipos pueden acceder. Y bueno, desde la Sociedad Argentina de Inmunología los esperamos en la próxima actividad, que creo que va a ser la última del año, en el webinar que nos dará el doctor Cosariza el próximo 4 de diciembre a la 1 del mediodía, hora argentina. Así que los esperamos. Muchas gracias a todos. Nos vemos muy pronto. Chao, gracias.